Hormones. Many different chemical species act as hormones. Steroid hormones, all derived from cholesterol, regulate metabolism, salt and water balances, inflammatory processes, and sexual function. Several hormones are amino acid derivatives. Among these are epinephrine and norepinephrine, which regulate smooth muscle contraction and relaxation, blood pressure, cardiac rate, and the processes of lipolysis and glycogenolysis, and the thyroid hormones, which stimulate metabolism. Peptide hormones are a large group of hormones that appear to regulate processes in all body tissues, including the release of yet other hormones. Hormones and other signal molecules in biological systems bind with very high affinities to the receptors, displaying Kd values in the range of 10 to the minus 12th to 10 to the minus 6 mole. The concentrations of hormones are maintained at levels equivalent to or slightly above these Kd values. Once hormonal effects have been induced, the hormone is usually rapidly metabolized. The steroid hormones include the glucocorticoids, the mineralocorticoids, vitamin D, and the sex hormones. The steroid hormones exert their effects in two ways. First, by entering cells and migrating to the nucleus, steroid hormones act as transcription regulators, modulating gene expression. These effects of the steroid hormones occur on timescales of hours and involve synthesis of new proteins. Considerable evidence has accumulated, however, that steroids can also act at the cell membrane, directly regulating ligand-gated ion channels and perhaps other processes. These latter processes take place very rapidly on timescales of seconds and minutes. The largest class of hormones in vertebrate organisms is that of the polypeptide hormones. One of the first polypeptide hormones to be discovered, insulin, was described by Frederick G. Banting and Charles Best in 1921. Insulin, which is typical of the secreted polypeptide hormones, is a secretion of the pancreas that controls glucose utilization and promotes the synthesis of proteins, fatty acids, and glycogen. Many other polypeptide hormones are produced and processed in a manner similar to that of insulin. Three unifying features of their synthesis and cellular processing should be noted. First, all secreted polypeptide hormones are originally synthesized with a signal sequence which facilitates their eventual direction to secretory granules and thence to the extracellular milieu. Second, peptide hormones are usually synthesized from mRNA as inactive precursors termed pre-prohormones, which become activated by proteolysis. Third, a single polypeptide precursor or pre-prohormone may produce several different peptide hormones by suitable proteolytic processing. The impressive example of the production of many hormone products from a single precursor is the case of pre-pro-opiomelanocortin, a 250 residue precursor peptide synthesized in the pituitary gland. A cascade of proteolytic steps produces, as the name implies, a natural opiate substance, endorphin, and several other hormones. Endorphins and other opiate hormones are produced by the body in response to systemic stress. These substances probably contribute to the runner's high that marathon runners describe. And those are hormones. Have a great day.